of the session is done by Dr. Prasun Mukherjee. Before the lecture starts, I would really want to let you know a little bit about the gentleman. Dr. Prasun Mukherjee worked as the head environmental biotechnology section and group leader, agriculture, microbiology, nuclear agriculture and biotechnology division at Bhabha Atomic Research Center, Mumbai. He obtained PhD in plant pathology in 1991 from GB Plant University of Agriculture and Technology. He was the university topper of BSc, MSc and PhD, and he has been a visiting scientist and scholar at the Israel Institute of Technology Haifa in Texas and A&M University and College Station. Dr. Mukherjee has been working on the biofungicides Trichoderma SPP for more than 30 years and has made seminal contributions in genetics and genomics of these fungi, especially in the area of signal transduction and secondary metabolism. He has also developed novel mass multiplication method for which a patent has been filed and the technology transferred to private entrepreneur. He has published more than 100 research papers and review articles in the highly reputed journal like Nature Microbiology Review, Genome Biology, Microbiology, BBRC, etc. with 8,621 citations. He has won several awards for his research and contribution. He is an elected fellow of the National Academy of Science, India, National Academy of Agricultural Science as well. He topped ARS examination in plant pathology, joined BARC. He reviewed Dr. K.S. Krishnan DAE Research Fellowship. And yes, the topic, the discussion of his is on a video mode. So he is not present over here, but let's just go through this now.
A very good morning to everybody. My apologies for not being able to come personally. So I'll be presenting my experience on Trichoderma, the most commonly used biofungicide. The title is Basic Biology Meets Applications. Basically, I'll be covering both the basic research which we have done and also some commercialization aspect. As you know, like pests and diseases, they cause like huge damage to crops. According to this report, like it's 10 to 40%, but in certain cases, like it could be 100% losses. Traditionally, chemical pesticides and fungicides have been in use for management of these diseases and pests, but due to their toxic effects and environmental side effects, many pesticides are getting banned, as you can see from this news article. Also here, many of them are fungicides, so basically our options are getting limited to control these diseases. So we have been targeting pathogens like Sclerotinia rot, C. Ragitria solani, which are very broad host range. Because they are very broad host range, it is very difficult or almost impossible to get disease-resistant plants. So either chemical or biological, these are two options, but they are like soil-borne in nature. So chemical control is also not very effective. These fungi produce resting structures which are called sclerotia. As you can see here, a close-up of a sclerosium. So these sclerosia, they survive in soil, and whenever they come in contact with the seeds or the root, they start infecting it. For these type of pathogen, especially biological control is a very good option. And as I mentioned earlier, like Trichoderma species are the most popular biofungicides. Trichoderma species have certain advantages, like they are omnipresent. There are more than 300 species recorded, but out of these 300, about 20 are frequently isolated from soil and rhizosphere. Most of the new species are found in forest, on deadwood, tree bark and mushrooms. And some species are true endophytes, like they live within the plant. They are used widely as plant disease biocontrol agents. In India alone, more than 300 products are available. They also promote plant growth. They are reported to control soil-borne foliar pathogens, fungal and bacterial pathogens, also nematodes, viruses, and in some cases insects also, because they can induce systemic resistance. Among the mechanisms of biocontrol, there are three very well-defined mechanisms. One is mycoparasitism, one is antibiosis, and one is induced plant defense. By mycoparasitism, we mean that when they come together, a trichoderma can parasitize other fungi, as you can see from this picture. The thick one is the pathogen, the thin one is trichoderma. So trichoderma can coil around and then lies the mycelium of the pathogen. They can also parasitize the resting structures, which basically survive in soil. So this is an additional advantage with these biocontrol agents. In this assay, you can see the sclerotia are getting colonized by trichoderma, green growth on the brown sclerotia. And when they are treated on a selective medium, the sclerotia cannot grow as compared to control. So that means the sclerotia have been killed by trichoderma. Antibiosis, like they're basically secondary metabolites which are produced by microbes. And trichoderma also produce a lot of secondary metabolites, and some of them are inhibitory to the pathogen, as you can see from this slide. And trichoderma variants, especially like which I work on, they produce viridin, hepchloric acid, cleovirin, and volatile sesquiterpenes. So we have been able to identify the genes for all these metabolites. I'll come to that. Trichoderma, volatile metabolites, they are very good promoters of plant growth, especially root growth, as you see in this picture from our own laboratory. Arabidopsis plants are exposed to the volatiles of trichoderma variants, and as compared to control, you can see the profuse growth of the root. There is no contact with trichoderma here, only the volatiles influence the roots to grow to such an extent. Trichoderma can colonize the roots, as you can see here in the left side, without trichoderma and with trichoderma, extensive growth on the root. They also can go inside the root. And when trichoderma is inside the root, they can induce resistance in the plant, like in the leaf also. So if we inoculate the leaf with a foliar pathogen, it's very likely that if the root is colonized by trichoderma, the disease reaction will be less compared to, the lesion size will be less compared to the non-inoculated control. So we have been working on all these three aspects, identifying the genes using knockout, knockdown and overexpression, the genes which are involved in mycoparasitism, secondary metabolism and induced resistance. I will give some examples from our studies. First, the mycoparasitism. So we have identified two genes. We have identified many genes actually. I am showing data for two. One is called PGOI gene, PGOI1 we call, and one is ECM33 gene. So you can see that wild type, it is overgrowing sclerotial ulcer. But if you remove this gene PGOI1 or ECM33, actually the pathogen is overgrowing trichoderma. So it has lost the mycoparasitic ability on sclerotial ulcer. Similarly, on Digitrona solanae also, if you can see here, like wild type has completely overgrown Digitrona solanae in 6 days, whether PGOI knockout mutant and ECM knockout mutant, they are yet to overgrow. They cannot overgrow basically, very slow on Digitrona solanae also. So, so this is the this is an example where we identify the genes which are basically involved in mycoparasitism. Induced defense also we have been working, especially the identification of the elicitor proteins or effector proteins. There is a study in Israel where they have done a secretome analysis of trichoderma when it is interacting with maize roots. So they have found one small secreted cysteine-rich protein which is upregulated. Actually 13 are downregulated and 1 was upregulated. So we targeted this upregulated cysteine-rich protein. This is the sequence of the protein. It has no conjugate domain, no structural ortholog when we studied. So what we did is we expressed this in E. coli, this protein, and then in solution it was characterized and it exists as a dimer. So we elucidated the structure of this effector protein, and surprisingly, even though there is no homology with any protein, known protein, the structurally it was very similar to ALT-A1, and PEVD1. So these proteins are ALTA1, you know, like this is an effector protein produced by alternaria, and PEVD1 by verticillium. So both are involved in pathogenesis. But so surprising, it was like 8 to 10% only sequence identity, but the structure it was very similar to these effector proteins known in plant pathogens. When we inoculated the root of maize with this protein, what we found is, and inoculated the leaf with Cochleobola heterostrophes, a necrotroph, what we found is it produces a bigger lesion size, which is not surprising because it's a necrotroph, and it induces salicylic acid pathway. So this particular protein induces salicylic acid pathway, and that's how the plants becoming more susceptible to necrotroph. We are testing if it is becoming resistant to biotroph and hemibiotrop. In terms of secondary metabolism, we have done again extensive studies. The objective was to identify the genes involved in secondary metabolism. So we started with mutation of Trichoderma, and we were lucky to get two mutants of Trichoderma. One is called G2 and one is M7. So G2 is an overproducing mutant for secondary metabolites, and M7 it does not produce or produces below detection limit of secondary metabolites. So M7 we have used as a genetic tool for unearthing the secondary metabolism related genes, and G2 we have used as a developed into a commercial product. So I'll come to that later. Most secondary metabolites are produced by gene clusters. What do you mean by gene clusters is like there are several genes which are together and they produce the code for the same metabolite. Different genes do different functions, and then as an example here, it is gliotoxin gene cluster, which is produced by Trichoderma virens, Q strains, and also by Aspergillus fumigator. So these gene clusters are very often horizontally transferred from one to the other. We have isolated or identified the gene cluster, which is a non-ribosomal peptide. It's a very strong antibiotic against oomycetes. So using gene knockout, whole genome sequencing followed by gene knockout, we have identified these metabolites, these gene clusters. And also we have identified the same way viridin gene cluster. So 21 genes, they are responsible for production of one compound, viridin. Heptanoic acid, again, it's a very important secondary metabolite. And interesting thing here is that we have found out that two gene clusters, independent gene clusters, they are responsible for production of heptanoic acid. So one cluster, it takes the terpene cyclase, it's a terpene, and other cluster, it takes the 3 cytochrome P450 monooxygenases to produce the compound. Siderophores are very important for competition for iron in the rhizosphere. So we have also been able to identify the siderophore gene clusters and also proved that they are producing siderophores by gene knockout. The V cluster is for volatiles. I told like Trichoderma produces both the volatile and non-volatile metabolites. So this is the cluster which is responsible for production of 22 volatile sesquiterpenes. We knocked out the V4 gene, which is a terpene place, and as you can see in this picture, the red one is the wild type and the blue one is the mutant, knockout mutant. So almost all the volatile metabolites are gone in the mutant just because we knocked out one gene. So this gene cluster was identified as a volatile metabolite producing gene cluster in Trichoderma virens. Now we have done the basic research and now time to discuss about the commercialization. As I told you earlier, we have a mutant which is called G2, which is producing more secondary metabolites. As you can see here, wild type filtrate color and G2 filtrate color. G2 filtrate color is darkened. If we add the filtrate to the medium, the growth of the pathogen is inhibited more by the mutant than wild type. Also in HPLC, we can see it's not a neat picture, but you can see it is producing more secondary metabolites. We did the transcriptome analysis also, wild type and G2 mutant. And again, we find like 20 genes per second metabolites production are upregulated in the mutant, and also there are several other genes which are also upregulated in the mutant. G2 is as good as a wild type in high parasitism, so we did the microparasitism study. You can see here, like, against sclerosis wild type and G2 mutant both are overgrowing and lysing the mycelium of sclerosis. For parasitism of sclerosia also, like we have did the assay, so wild type and G2 both are equally effective. After knowing that G2 is producing more secondary metabolites and is as effective as the wild type in mycoparasitism, so we tried to formulate it. We looked for different seed, different grains. Traditionally, jowar is used, but jowar is expensive, so we used tamarind seeds as a mass multiplication medium. And tamarind seed has an added advantage because they are very sticky, so you don't have to add any sticker in the formulation. So this was formulated as a talcum powder formulation, and it produces about 2 × 10⁸ CFU per gram. Very easy to coat the seeds with this formulation. You take the seed, little bit water, and then put the formulation required amount. We recommend 5 to 10 gram per kg seeds, and here it is 5 gram per kg seeds, and still you see the seeds are beautifully coated. We did a lot of studies in pot and small plots, wild type and mutant comparison, seed treatment. So here one example where Sclerotinia rhodopsii was added in a huge amount quantity, so that we get almost 100% mortality. In that disease pressure also, you can see the G2 is performing better than the wild type in controlling the pathogen Sclerotinia rhodopsii. We took up a trial to begin with in 2015-16 in Raipur. My friend Dr. Cuttack from IGKV Raipur, he was interested to study this test in the field. And then we put in a small plot and then see the effect, like seed treatment with Tricho, BARC, gave the name, and control. Huge difference. This encouraged us to take up this further and then take it to the commercialization scale level. Then again, 2018-19, we took up a trial. This plot was not infested much with the polar rot pathogen, but the growth promotion effect was very much visible here. And in farmers' field, we took up trials in many farmers' field over years. This is one example, like where the yield gain was 4 quintal per hectare, which translates to about 20,000 rupees benefit per hectare. Seed treatment costs very little, like 100 rupees or so. ICAR got interested to study this in AICRP, chickpea in multi-location trials. So 17-18, 18-19 we have taken the trial, and here also you can see, like on an average, Tricho BARC formulation gave better control than other isolates. So that I have not included that data because that can be seen in the next slides. But you see, like the huge difference between control and the treated. So ICAR continued this trial for another 2 years. 2021, they've taken the trial in 7 locations, and along with our formulation, 3 other formulations are included. And you can see here, like if you see the average, total average, like overall performance, our formulation was the best, and the average yield gain was 63% over control. Similarly, again 21-22, it was extended further to 8 locations, and then many other Trichoderma strains were included. Again, here also it was showing the superior overall performance, and here yield gain over control was about 73. So, encouraged by this, a frontline trial was also taken up in MP, FLD. So here again you see like 24% increase in the yield. So that is based on these data, ICR recommended seed treatment with trichobark for chickpea cultivation in 2022 meeting. We continue to take the trial in farmers field. Like 24 farmers were covered in this trial. Again, the average you see like control and treated, 24% increase, so it was 2.9 quintal per hectare, which again translates to about 15,000 rupees per hectare net profit. This was covered in a local newspaper in Chhattisgarh. And another trial in the farmer. We tried in soybean also in KVK, and again here we found that in soybean also it is giving a very good yield, as you can see here. So here about 3 quintal per hectare enhancement was there, and it translates to about 11,000 rupees benefit per hectare. In 2020, again we have taken up the trial. Again we found the similar result. 4.2 quintal per hectare was the additional gain in yield, which translates to more than 60,000 rupees per hectare profit. In lentil, we have taken up extensively the trials in farmers field in West Bengal. In this picture, you can see like 3 years, 3 seasons. So average of 30 farmers field in 2 years, and then third year 10 farmers field. Again, taking all the averages, it is about 17,000 rupees per hectare profit. You can see a control and treated plot. The growth was robust, which we extended to the next year also, 21-22. Again 10 farmers. So here it is very like big difference we got, like 674 kg per hectare, which translates to 37,000 rupees per hectare, and this is average of 10 farmers field trap. You can see the plot here, like treated, and T4 is control, the right side is treated, seed treatment. Very apparent, like the robust growth due to the seed treatment with trichoderma. We took up the trial in paddy also. Again, Dr. Kotastana was interested. He basically works on paddy. I know a lot of work is going on in NRI, Cuttack, like very excellent work on Trichoderma application in paddy for growth promotion and disease control. For our formulation also we tried in transplanted paddy where root deep method was used, and you can see here, like again we had like about 21% improvement in the yield, which was attributed to the increasing number of till. We tried in cumin in Jodhpur. It's a cash crop there, so we thought it is very interesting to know if it is working in Rajasthan, which is a very different type of soil and environment. So it was found to be very good in Jodhpur also, in cumin. Other crops also like guar and chickpea, I'm not showing those. This formulation has very good growth promotion effect. You can see here in maize, again in Chhattisgarh, control versus treated, very clear. In spinach, Dr. Kotastane wanted to take up a trial in spinach and then see seed treatment improve the seedling stand, because the soil having damping of issue. So you can see here the stand between treated and control, a huge difference. In Navsari, Gujarat, again a damping of infested brinjal nursery, we took up a trial. So you can see the control versus treated. Both the growth promotion and plant stand was excellent in the seed treatment. Under conditions, field condition, other vegetables also it was tried, and then everywhere we are getting like bigger biomass, greater biomass in vegetables. Again, Dr. Kotas, thanks to him. So after all these data, we had like confidence that yes, this is working in the field. So next challenge was to register this with the CIB-NRC, which is very challenging. Most people stop there. But fortunately, we had a good support of our institute and then other places, and of course ICAR. So we could proceed with a register. As you know, it's a very complicated process. We need a lot of data, including like formulation, nature of formulation, then fingerprinting data, and then biological characteristics and chemistry of the product, different tests we have to perform, whether there is a contamination or not, then bioefficacy data, of course, field data, laboratory data, toxicity data for both mother culture and formulation. It's very expensive, but we could do it. And then mammalian toxicity, environmental safety testing like mammals, bird, fish, invertebrates, like earthworm. So all these tests, packaging and labeling, all these generated, and then finally we could get it registered as a biopesticide. It was the registration was done by Mrs. Mbio Limited. This is the first mutant microbe to be registered for field application, and then the product has been launched already. It is called Viren. This technology has been transferred to 5 companies so far, including Patanjali and UPL. UPL is a very big company, so is Patanjali. So we are hoping that many more products based on this technology will in the market, come out in the market. Those who are interested to read some of the publications can visit my Google Scholar page or write an email to me. I will be very happy to. I acknowledge my collaborators, especially Professor Kotasthane. I told you many times, like he is the person who drove this technology to the field. Professor Rajiv Nath from BCKB, Dr. Saxena, Dr. Nilima Karmakar, Dr. Manish Kumar from Jodhpur, she is from Gujarat, Navsari, and my students and colleagues, Ravindra Bansal, Sharkane and Sikha Pachori, of course BRC and Department of Veterinary. Thank you so much.
So that was from Dr. Prasun Mukherjee. And now we'll move towards the next by Professor Rob. Is he there? Online? Sir? Okay, any questions? Any questions for Sir? Okay, so seems like that was very clear. So probably no questions for now. Thank you so much, sir, for joining online as well.
Thank you.
Thank you, sir. Okay, so can we now proceed? Yes. Okay. The next lecture is by Professor Robert Henry, University of Queensland, Brisbane, Australia. He'll be talking on the topic genetic improvement of crops to balance the needs of food security and climate change. We'll connect him on Zoom call.
Good morning.
Good morning.
Today I'm going to talk about genetic improvement of crops to balance the needs of food security and climate change. The roles that agriculture can play in addressing climate change are many. One of the things we often do is try to adapt agriculture to climate change, and that's been an immediate response that's been underway for a long time. More recently, people are looking to use climate change or use agriculture to mitigate climate change by reducing emissions of carbon and greenhouse gases in agricultural production. I wanted to talk about the balance of those with a third strategy or a third important area, which is using agriculture to reduce carbon emissions in industries outside of agriculture. So in this, we're talking about using agriculture to produce renewable sources of carbon that can be used to replace oil for the production of not only biofuels but bioplastics and chemicals, all of the things that we currently produce from oil. And this is particularly important for biomaterials, where we can't use things like solar and wind. We can do that for energy uses of oil, but not for the non-energy uses where we actually need carbon compounds. So agriculture is the crucial industry for addressing climate change in this aspect. And back in 2010, I wrote a book that really stressed the way in which food production and food security from agriculture needs to be balanced with the conservation of the biodiversity on which agriculture depends, and with the production of non-food products from agriculture. All three of these things are essential. We can't just conserve biodiversity or just produce food, or in fact just produce non-food products, but equally they all are required. So what we need is to be efficient, particularly in food, but in the non-food production from agriculture, so that it doesn't compete significantly or to any extent with food production. So the way that we prefer to do this is to produce particularly those biomaterials that represent the main products that are made today from oil. Energy is actually in minor use, although we have for some time been producing ethanol and biodiesel. Into the future, the things that will remain that need the use of agriculture will be long-distance flight, so we need sustainable aviation fuel from agriculture, and we need those platform chemicals that we use to produce plastics, carpets, fabrics, all of the things that we currently produce from petrochemical sources. And the favoured path to achieving this is what we call the biochemical route. In this path, we take the non-edible parts of plants, the residues and biomass that doesn't have immediate human food use. Using enzymes, we produce sugars, which we then, using microbial approaches, convert to the chemicals that we need. And this provides a path that replaces the refining of oil. One of the critical applications of this technology is the conversion of biomass to fuels, particularly jet fuel, and here we're depicting a path to dimethyl cyclooctane. This is a superior molecule, better than Jet A. Planes can fly further using this product. So we're talking about superior products derived directly from agriculture. And the need for these products will remain with us for some time. We're going to see aeroplanes become electric just as cars are, and but this will allow regional flights to be electrified in the relatively near future, as you can see on this timeline here. We'll start to see that already by 2025, and this will be widespread. By 2040, even short-haul flights and medium-haul flights may eventually become electrified, but the long-haul flights of several hours are going to continue to require a more concentrated energy source, a liquid fuel into the future, even beyond 2050, with all of the technologies we're aware of at the moment. And so this needs to be sourced from agriculture. If we're going to reach net zero carbon emissions by 2050, we need to produce jet fuel for long haul from agriculture. The two preferred crops that we work with in this regard are sugarcane and eucalypts. One is an example of a grassy biomass, and there are many other grasses that are also candidates for this role. And the eucalypts are an example of woody biomass. Woody biomass has an advantage in that it can be stored longer than the grass-based biomass. And the reason that we focus on species like this is that they're highly productive. If we're going to avoid competition with food production, we need to only focus on the production of very fast-growing, very highly productive biomass. These are both species where 100 tonnes of biomatter per hectare per annum is achievable. In fact, 200 tonnes is now routinely achieved. Species that have significantly lower biomass potential really do risk the need to plant such large areas that they will actually compete with food production. And the technologies that we have that really drive the innovation here are the advances in genome sequencing, genome technology, and the advances in gene editing, allowing us to modify these plants to make them suitable for this use. Gene editing, we now have become routine in doing in the grasses, and so this is a way that we can modify the composition of the biomass to make these processes more efficient. We're also using high throughput mutagenesis. We've developed a platform in sorghum. We have 150,000 mutants of sorghum developed in collaboration with the Carlsberg research laboratories. And this population allows us to very quickly, in a matter of days, find a mutation in any gene and phenotype it. And this is a way in which we can test very large numbers of genes to find the ones that will give us the attributes that we want. The advances in DNA sequencing that we've made allow exact gene modification, because now we have an almost perfect gene sequence that we can work with. We can avoid non-target changes. We can make quite deliberately the changes in the plant that we want. We've applied these advances, and here's some work by Adam Healy's efforts, where we've achieved a full sequence of the polyploid sugarcane. This is the last of the 20 major crops in the world to have a genome sequence. It's very complex, but it's now possible with the advances in technology that we have. And this means through the advances documented in the papers listed here, we've compared all the long-read sequencing techniques, we've looked at improvements in the chemistry and improvements in the software available for the assembly and analysis of these genomes. And in the last 2 years particularly, we've seen dramatic advances in those areas, which mean that this can become much cheaper and much more routine. We've got a number of reviews that have followed this advance. What we've done for sugarcane is come up with high-quality reference transcriptomes using long-read sequencing and various other techniques that mean we now know of all the genes that are expressed in the plant, and this allows us to better target efforts to manipulate them. We've also got a greater understanding about how the composition of the biomass influences the